Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing University of Hyderabad Integrated MSc PhD Entrance Examination for the subject Biotechnology conducted in the year 2018. I will be discussing only the biology part. If you are new to my channel, please subscribe. Answer. The first question. The Nobel Prize in Physiology and Medicine 2017 was awarded to Jeffrey C. Hall, Michael Rauschenbach, and Michael W. Young for their work on discoveries of molecular mechanism controlling circadian rhythm. Answer is option C. A disease always present in a population is referred to as it is endemic. Answer is option B. Disinfectant agents to clean surgical wounds in order to control infections in humans are introduced by. So they were introduced by option D, that is Lister. Then Earth's atmosphere layer are in the order by the distance from the sea level. So it is troposphere, less than stratosphere, then mesosphere, thermosphere and exosphere. Answer is option D. Oxidative phosphorylation involves simultaneous oxidation and phosphorylation to finally form ATP. Answer is option B. Then total charges on the peptide RREEA is. So this is the peptide, that is RR represent arginine, E represent glutamic acid and A represent alanine. So this is the peptide sequence, and you know that it has got arginine has got side chain pKa value of 12.5 and glutamic acid is having side chain pKa value of 4. At the ends it is 2 and 10. So when pH is greater than pKa, it is deprotonated and you get a negative charge. And when pH is less than pKa, it is protonated and you get a positive charge. Here the pH is 7, physiological pH. So pH is less than 12.5, so you get a positive charge over there. Similarly over here also positive charge and here also positive charge. Now 7 is greater than pKa of 4 and 2, so you get a negative charge over here. So 1 plus 1 plus 1 plus minus 1 plus minus 1 plus minus 1, that will be equal to 0. So the answer here is option D, that is none of the above. Then which of the following amino acid has more than one chiral center? So of these, threonine is having more than one chiral center, that is at carbon number 1 and carbon number 2. So the answer here is option C. Then the growth of a facultative anaerobe will be. So a facultative or aerotolerant anaerobes can grow throughout the medium but will primarily grow in the middle of the tube between the oxygen rich and oxygen free zone. So the answer here is at the middle of the culture medium. Answer is option C. The tough water insoluble protein present in the outer portion of the epidermis of skin is keratin. Answer is option C. Then which of the following is a part of a virus? So it is capsid, core, nucleocapsid, all of the above. Answer is option D, that is all of the above. Then if both circular and open circular DNAs are analyzed in a single gel electrophoresis, agarose gel electrophoresis, then both the DNAs migrate at same speed. Circular migrates faster. Circular will migrate faster. That is the answer. Because circular is, it means supercoiled. So answer is option B, that is circular DNA migrates faster. Then in the case of edible vaccines, the antigen is available as. So edible vaccines are subunit vaccine that contain antigen proteins for a pathogen but lack the genes for the full pathogen to form. So they are expressed in the vegetable or the fruit while they are growing. Answer is option C. Then antigen presenting cells that activate helper T cells must express which one of the following on their surfaces? So they must express class 2 MHC antigens. Answer is option D. While working with a mutant DNA polymerase that is partially defective, The in vitro experiments show an error rate of 10 raised to minus 3 as compared to an expected error rate of 10 raised to minus 6. Which of the following activity is the mutant polymerase likely to be missing as compared to the normal polymerase? So it is the 3' 5' exonuclease activity. Answer is option C. The 3' 5' exonuclease activity allow a polymerase to remove misincorporated nucleotides, and this ensures the high fidelity DNA sequences that is required for faithful replication. So it is lacking 3' 5' exonuclease activity. Then the association between the ribosome and the messenger RNA molecule occurs in the cytoplasm. That is answer C. What kind of proteins are recognized by proteasome? So they are ubiquitinated proteins. Answer is option. Then the role of HSP60 in protein folding. So to provide sequestered chamber where protein folding can occur without interference from surrounding molecules. So answer is option C. Then which of the following protein is required for one round of in vitro DNA replication using a single-stranded linear DNA as template and the appropriate DNA primer? So they have asked which of the following protein is required for one round of in vitro DNA replication using a single-stranded DNA as the template and appropriate DNA primer is provided. Now primase is not needed because DNA primer is provided. Then ligase is not needed, it is for joining DNA. Helicase is for unwinding the helix. We are provided with single-stranded DNA, so that also is not needed. So we require only DNA polymerase. Answer is option B. Then how many distinct transfer RNA molecules are expected to be present in a cell, not including those present in the mitochondria? So between 20 and 61. Why? Because to achieve fidelity of translation, there must be a recognizably different tRNA for each amino acid, giving a maximum number of 20. However, most amino acids are coded by more than one codon, and a different tRNA could exist for each codon, giving 61 if each codon specifying an amino acid has a different tRNA, or 64 if the three stop message codons have tRNAs. In fact, because the third base of a codon is usually not recognized exactly, some tRNA anticodon can recognize more than one codon. This means that the actual number of tRNA is less than 61. However, many amino acids have more than one tRNA. So the number of separate ones is greater than 20. So the answer will be between 20 and 61. Then next one. Which among the following protein is involved in peptide initiation and chain elongation is GT-base switch? So both EFTU and EFG. Answer is option C. The major difference between prokaryotic and eukaryotic protein synthesis mechanisms are in. So the initiation of synthesis. Answer is option A. Then which of the following best describes cap modification of eukaryotic mRNA? A modified guanine nucleotide added to the 5' end of the transcript. Answer is option B. Then a phosphoglyceride is always. So it is always only a saturated or unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached. So the answer here is option B. The next one. Which of the following pairs is wrongly matched? So fruit juice pectinase, detergent lipase, alcohol nitrogenase, textile amylase, alcohol nitrogenase. So the question is related to the industrial application of enzyme. So alcohol is not produced by using nitrogenase. Nitrogenase perform complex function of nitrogen fixation by bacteria, and alcohol is produced by fermentation process of sugars carried out by the yeast. So the answer is option C. Then which of the following is not required for any of the techniques of DNA fingerprinting available at present? So zinc finger analysis. Answer is option C. Then in glycolysis during the oxidation, electrons are removed by, so it is by NAD+. Answer is option C. Then link between glycolysis Krebs cycle and beta oxidation of fatty acid is, it is acetyl-CoA. So the end product of glycolysis, that is pyruvic acid, is converted to acetyl-CoA by the process of oxidative decarboxylation in the presence of pyruvate dehydrogenase enzyme. And this acetyl-CoA, it enters the Krebs cycle where it is oxidized to produce energy in the form of ATP. Then beta oxidation of fatty acid also produce acetyl-CoA, which enters the Krebs cycle and produce energy. So acetyl-CoA, it plays a link between glycolysis, Krebs cycle and beta oxidation of fatty acids. Then which of the following processes are sensitive to extracellular DNases? So it is transformation. Answer is option A. Then what would happen if a gene coding a polypeptide of 50 amino acids, 25th codon UAC is mutated to UAA? So the polypeptide of 24 amino acids will be formed. Answer is option A, because UAA are termination codon and they do not code for any amino acid. Okay, and if in a gene encoding a polypeptide of 50 amino acid, 25th codon is mutated to UAA, then the chain will be terminated at that place because it becomes difficult for the RNA to bring amino acid from the amino acid pool. So in that case, the polypeptide of 24 amino acids will be formed. Then after mating with an HFR strain, what happens to an F- cell? This I have already discussed in one of the previous question papers. I don't know which one, but anyway, it stays F-, but picks some chromosomal genes. Because conjugation is typically disrupted before the fertility factor can be transferred. So the F- cell, it will remain F- itself, but it picks some chromosomal genes. So the answer is option D. Then which is the most common anaerobe in the normal body flora? So it is Escherichia coli. Answer is option D. Then the value of omega angle in transpeptide unit is only plus 180. Answer is option A. Then signals required for T cell activation. So peptide MHC complex, co-stimulation, cytokinins and other effector molecules. All of the above. This is a repeated question. Rapid but non-antigen specific immune response are produced by. So it is produced by the innate immune system. This is also a repeated question. Then next one. Which of the following are true with regards to interferons? So interferons, they inhibit virus replication by infected cell. Answer is option D. Then cells that release histamine and other vasoactive substances in response to allergens are. So they are the mast cells. Answer is option D. The next one. Which of the following disease is not an autoimmune disease? So bovine spongiform encephalitis, it is caused by a prion. So it is not an autoimmune disease. The T cell receptor can bind to antigenic peptides only when loaded onto MHC molecules. So answer is option B. Cytotoxic T cells are distinguished from helper T cells by the presence of CD8. Answer is option C. Then which of the following types of genes are not known in any mitochondrial genome? That is glycolytic genes. Glycolysis takes place in the cytoplasm. This is a similar question was asked in previous years. Okay, I don't know which question paper, but it was asked. Dash may contain the nitrogenous base uracil. So transfer and messenger RNA only. Answer is option D. DNA does not contain uracil. Then the Maxim Gilbert method of nucleotide sequencing is based on. It is based on chemical modification of DNA. This is also a repeated. Then next one. Which of one of the following is not a mechanism whereby B cells or antibodies contribute to immunity? So that is direct cell killing. Answer is option D. Then which of the following is a prime example of a ribonucleoprotein enzyme, the catalytic activity of which under non-physiological conditions resides solely in its RNA component? So it is RNase P. Answer is option C. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.